This is for Biology 223 Genetics. I wanted to go through some of our current knowledge of SARS-CoV-2, the COVID-19 disease-causing virus. I'm not an expert in these kind of viruses. The closest viruses similarity, what's relevant probably is work with retroviruses and with Atoka-Viridae system and some knowledge and many seminars picked up while working in virology and immunology, as well as molecular biology and structural biology. So I'm going to use this review, SARS and MERS, Recent Insights into Emerging Coronaviruses. This is published in Nature's Reviews Microbiology 2016, so predating this SARS-2. And you may recall, students wouldn't be old enough to recall SARS, but MERS has been more recent. In 2012, there's an outbreak, and it's endemic in camels in this part of the world. So I wanted to focus on the information, the molecular and structural biology, what we know about these kind of viruses based on the figure 2, which shows the physical structure, the genetic structure, and the life cycle. The life cycle is really the key to an overall understanding. There are many genetic issues I want to focus on here, and there, of course, in the disease, and this review is good because it talks about the disease as a whole, including how it's spread, and a great deal of what makes why virology is often part of microbiology and immunology, why its relationship to immunology is how the virus interacts with host immune system. That's beyond the scope of what I'm capable of discussing properly. However, the life cycle and the structural biology and the genetics, we can relate to a lot of things that come up in our class. So I don't have a color version here. Let's first go over the physical structure. So this is a membrane-enveloped virus. Not all viruses are membrane-enveloped. Retroviruses like HIV are membrane-enveloped. Polio is not. It has a lipid bilayer that's taken from the host, and then it has viral proteins, including, most importantly for us, the spike protein. The spike protein is very interesting because it's the protein by which the virus both binds to the host cell, the recipient cell, to facilitate entry, and the protein by which it fuses with the cell membranes and enters the cell. So it contains both the receptor binding and the fusion machinery. It's also the target of the immune system in that it neutralizing antibodies of the host then is recognize the spike protein and neutralize it. There's also the possibility of antibodies triggering the conformational change necessary for fusion and inactivating it, but I haven't seen any detailed information on that. Importantly, antibodies from mice or people from SARS and now from this SARS-CoV-2, the antisera from patients protects infected patients from the disease by neutralizing the particles. So now, otherwise, the other aspects, there's other proteins. It has a shell inside. It's a single-stranded RNA. It's a positive single-stranded RNA virus. Positive single-stranded RNA virus means it's made as a positive strand of RNA. Its genome is a positive strand, meaning that it's sense, meaning that it can be translated directly. And so in its life cycle, we'll go through that, there has to be a negative strand intermediate. There's no DNA intermediate. And what's one of the aspects of these kind of infectious agents is how clever they are, how sophisticated they are, how much regulatory mechanisms they employ. And that's a lot of molecular biology. And then beyond that, the ways in which they manipulate the host immune response is particularly complex, though not necessarily well understood, because this virus has not been studied really as much as some other viruses like influenza or HIV or polio. So the genome here is shown, it's a single RNA, positive sense, and it's shown here in this breakup, and we have some symbols here. We have blocks for open reading frames, and then we have displacement here. In this case, this indicates, so this is a negative 1 ribosomal frame shift site, and what that means is when this transcript, this mRNA is translated, the ribosome translates a polyprotein. It's a polyprotein that will create a polypeptide that's cleaved by the viral protease. We're going to distinguish between viral protease and host protease into individual proteins that have different function. Those are here. This 3 and 5 here are proteases of the virus. And then some fraction of the time, the ribosome stops, and some fraction of the time, it goes through a -1 frame shift and continues. So that's a way for viruses, HIV retroviruses do this, to regulate the stoichiometry of the proteins made. Because a polyprotein, then we have 1:1:1:1 stoichiometry, a frame shift, it can be adjusted. And conceivably, depending on the gene expression program of the virus, because it can have, I can tell you from lambda phage, which we discuss in the book, immediate early, delayed early, we have different programs. So it has these open reading frame, ORF1A and 1B, that go on to make some of the important viral proteins, including here, that's an RNA-dependent RNA polymerase. It's a replicase. And it's a replicase, that is, it is the polymerase that copies the viral genome into a negative sense and then back into full-length sense. And so that's replication. So this is the obvious, very important target. Unfortunately, the obvious drugs to replicases, things like ribavirin, which can have complex mechanism of action. One of the important ones by which it works against things like polio is that it causes the polymerase to lose fidelity and make more mutations, and the progeny then have reduced function. However, this class of RNA viruses, you may hear that RNA viruses mutate fast. Indeed, they do, because RNA polymerases don't have proofreading function. However, it does have a proofreading function through this ORF14, this non-structural protein 14 exonuclease. And so it's not clear whether typical nucleoside drugs that would work against other RNA viruses would work here. And also, the presence of this proofreading capability means that the RNA virus can have a larger genome than other RNA viruses, because RNA genome size is limited by the fidelity. The lower the fidelity of the copying, the replication, the shorter the genome. And so in organisms like us, we have very sophisticated replicative polymerases and proofreading and repair mechanisms, and some viruses don't. So polio, maybe it's 8,000 nucleotides. Then we have we have other genes here, including the structural genes. Those are the ones that appear in the virus, and other proteins, some of which I think have unknown function, and even ones that have known function presumably have other functions that are unknown. And a lot of the functions of these non-structural proteins can be to interact with the cellular machinery to regulate the virus production, but also interact with the host immune system for evasion, and that's what makes that's a large part of the pathogenesis of a virus, is how it manipulates the immune system. And in the case of SARS-CoV-2, there are thoughts that the problem, the pathogenesis, is because, at least this is true for MERS, so for MERS it's considered likely for the COVID-19, is that patients are not switching from their innate immune system to their adaptive immune response, from an innate immune response, such as interferon, interleukin-1, fevers, this sort of stuff, cytokines, to adaptive immune system, in which produces antibodies, and antibodies are the ones that can neutralize the virus. Let's go through the life cycle starting here. So the virus can attach via its spike protein. The spike protein is importantly has a cleavage site. It's called S1/S2, and what seems to be different in the case of this SARS-2 is that it has a mutation, which means that the cleavage site here has been mutated, so it's a very good site for a common host protease furin, and that may happen, in this case, before release. It may happen sometime before release, so that S1/S2 cleavage may happen. Then there's another cleavage, S2 prime, that appears to happen, at least in MERS, and it's thought to happen after entry to into the endosome. So the spike protein goes through this maturation, or so it can attach to the receptor and unmask its fusion machinery, which then get activated by this second host protease caused cleavage. So we have a very major difference from SARS in that there's this S1/S2 cleavage site is now very good. Also, the spike protein seems to have, some reports show that it has a higher affinity for the receptor, which is angiotensin converting enzyme 2, and that may relate to its disease capabilities or how pathogenic it is. So that's not so clear, and it's the furin cleavage site in the spike, which seems to be the most obvious difference. So the virus is then internalized into endosome. Endosome is acidified. At some point, the virus undergoes a spike protein undergoes a conformational change and fuses. It's similar to influenza, which is the prototype of understanding class 1 fusion proteins and how they enter. Then the genome can be translated to make the first set polyproteins here, which then include the replicase, which can make a negative strand. It does something quite interesting. It does its replication in these double membrane vesicles, and this escapes the virus, hides the virus from innate immune system sensors that recognize alien substances like double-stranded RNA. I'm going to stop here and start a second